welcome to the Deep Dive. Ready to explore some really cool developments. Today it's biomedicine and bioinformatics. We've got news articles, scientific publications, the whole shebang.
Sounds like a fascinating mix. Cancer research, regenerative medicine, new RNA analysis techniques. Where do we even begin?
How about with pancreatic cancer? News Medical is reporting on this new immune map. It's giving us some valuable clues about this aggressive disease.
It's pretty incredible. Researchers have been analyzing pancreatic cancer cells and how they make the immune system react. They created this map, and it's revealing a ton of details, like how the immune system is interacting with the tumor.
So basically they're mapping out the battlefield?
Exactly. And that could help us figure out new ways to fight back, new therapies,
That's a huge step forward, especially with how tough pancreatic cancer is to treat.
Oh, absolutely. If we understand how the tumor and the immune system are interacting, we can find ways to help the immune system fight harder or stop the tumor from hiding.
Okay, so switching gears a bit, let's talk about something amazing. News Medical has another article about how newborn babies can regenerate heart tissue after an injury.
Isn't that wild? It's because of their developing immune systems. When an adult heart gets injured, it scars. But a newborn's heart, it can actually regenerate the damaged tissue. No lasting damage.
Wow. So could we use this to help adults regenerate heart tissue after, say, a heart attack?
Maybe. Researchers are digging into exactly how this works, hoping to unlock the secrets of heart regeneration. And get this, the article also mentions pythons. They have similar regeneration abilities.
Pythons, seriously.
Seriously. Nature's full of surprises. Who knows what other healing secrets are out there, just waiting to be discovered. But for now, let's talk about something a bit more serious, tuberculosis. It's still a major global health problem, taking millions of lives each year.
It's a tough battle, that's for sure.
Yeah, it is. But there's good news. News Medical is reporting on some groundbreaking advances in tuberculosis vaccines and therapies.
That's encouraging. Any details?
Unfortunately, no. The article was pretty vague on specifics. But hey, progress is progress, right? Even without the nitty-gritty, it's a little glimmer of hope.
I agree. Every step forward counts. Okay, ready for a deep dive into the world of RNA. Often overshadowed by DNA, but it's a pretty fascinating molecule in its own right.
Absolutely. And the RNA-seq blog has a whole bunch of articles about it. They really highlight how multifaceted RNA is.
So it's more than just the messenger between DNA and proteins.
Way more. It's crucial for regulating gene expression. Scientists are developing these super sophisticated tools to understand exactly what it does.
Like what?
Well, there's this technique called TaDRIM-seq. It lets researchers study something called chromatin-associated RNAs and how they interact with DNA.
Chromatin. Okay, you're going to have to break that down for me. It sounds a bit complicated.
Think of it like this DNA is a huge library of genes. And RNAs, they're like the librarians deciding which genes get accessed. TaDRIM-seq lets us spy on those librarians, figure out how they control the flow of genetic information.
Okay, I get it now. That's a pretty cool analogy. So how does this help us with diseases?
Well, if we can understand how RNAs work, we could target them with therapies. Imagine being able to correct faulty gene expression at its source. That's powerful stuff. And that's not all. The RNA-seq blog has articles about using RNA sequencing to figure out drug resistance in lung cancer patients and to understand tumor diversity in Waldenstrom's macroglobulinemia.
Wow, so RNA is really having a moment in the research spotlight. What else?
Oh, there's more. They're also talking about spatial transcriptomics. It's a way to visualize tissue architecture in a whole new way. Basically, it lets you map where different RNA molecules are and what they're doing within a tissue sample. Pretty amazing, right?
Yeah, that's incredible. It sounds like RNA research is opening up all sorts of possibilities for personalized medicine and treating diseases in a whole new way.
Absolutely. We're just scratching the surface of what RNA can do. Okay, let's get back to those amazing regenerating pythons. Remember we talked about their hearts?
Yeah, how could I forget?
Well, it turns out their regeneration powers don't stop there. An article from the RNA-seq blog talks about how they can regenerate their intestines after a big meal.
Really? How does that even work?
Researchers used single-nucleus RNA sequencing to figure out the genes and pathways involved. It's pretty mind-blowing.
So pythons can regenerate hearts and intestines. What can't they do? Do you think this research could help with human digestive disorders?
Possibly. Imagine if we could stimulate intestinal regeneration in people with Crohn's disease or ulcerative colitis. That would be a game changer.
It's amazing what we can learn from the animal kingdom.
It really is. Nature is full of incredible adaptations. We just have to pay attention. You know, it's really interesting. Those articles from nature.com, a lot of them didn't have summaries, so we're kind of going in blind here.
Ooh, I love a good mystery. What have we got?
The first one is called Insights into Genetics of Floral Development in Ambarella trichopoda bale through genome-wide survey and expression analysis of MADS box transcription factors.
Okay, whoa, that's a mouthful. Can you translate that into English for us non-botanists?
All right, so Ambarella trichopoda It's a really special plant, like a living fossil, represents the earliest flowering plants. And MADS box transcription factors. They control a lot of things in how plants develop, especially flowers.
So it's about how flowers evolved.
Exactly. It's basic botany, but really important stuff.
Cool. What's next?
Okay, this one's called On Board with Comet to Improve Omics Prediction Models.
Omics, I feel like I've heard that before, but I'm drawing a blank.
Omics is like a catch-all term. It means studying lots of biological molecules at once. Genomics is for DNA, proteomics is for proteins, things like that. And prediction models, they're used all over to try to guess what will happen, like will someone get a disease? Will a treatment work?
So this COMET thing, is it making those models better?
Seems like it. Could be a big deal for personalized medicine. Doctors could make much better decisions if they had really accurate predictions based on your unique biology. Okay, how about this one? Author correction, Interformer, an interaction-aware model for protein-ligand docking and affinity prediction.
Author correction. Sounds boring.
Not really. The original article is about how proteins and ligands fit together. Ligands can be drugs or other molecules that attach to proteins.
Right, and that can turn things on or off in the body.
Exactly. And this Interformer thing, it's a computer model that predicts how strong that connection is, how well they stick together.
So useful for developing new drugs.
Super useful. They can test out drug candidates virtually and see which ones are most likely to bind to their target. No more trial and error in the lab.
That would speed things up a lot. What else?
This one is about cells. Automated cytometric gating with human-level performance using bivariate segmentation.
Psychometry. Isn't that analyzing cells?
Yep. And they often need to separate different types of cells. That's called gating. Sounds like this is a fancy way to automate it.
Wow. No more scientists hunched over microscopes, sorting cells by hand.
Haha, right. Automation is the future, my friend, especially when you're dealing with tons of data. Okay, get ready for this one. Segment anything for microscopy.
Sounds kind of familiar. Didn't we just talk about spatial transcriptomics? That was about looking at tissue, right?
You got it. Well, microscopy is another way to visualize cells and tissues. And segmentation means you're outlining specific parts within those images. Sounds like segment anything is an algorithm that can do it automatically.
Wow. So no more tracing things by hand.
If this lives up to its name, yep, this could be huge for all sorts of things. Research, diagnosing diseases. Imagine if we could automatically pick up on tiny changes in cells really early on.
That's wild.
And speaking of analyzing images, check this out. CellPose 3. One-click image restoration for improved cellular segmentation.
CellPose 3, what's that?
Sounds like a software program. Uses AI to make microscopic images better so it's easier to see those individual cells and segment them. And the one-click part, that means it's probably super easy to use, even if you're not a computer whiz.
Perfect for researchers who just want to get to the science. Okay, what else?
This one has a pretty interesting title, Genomic Data Sharing. You don't know what you've got till it's gone.
Ominous.
It makes you think, right? Genomic data is super valuable for research, but sharing it safely, protecting people's privacy, that's a tough challenge.
I get it. You want to be open and collaborative, but you also have to be responsible.
Exactly. And maybe this article talks about how to find that balance. Good policies, good systems for sharing data without putting people at risk.
Makes sense. All right, what's next?
Back to cancer research. ZCCHC4 regulates esophageal cancer progression and cisplatin resistance through ASME access.
Wow. Okay, I'm lost already. Break it down for me.
ZCCHC4 is a protein. They're looking at its role in esophageal cancer. Cisplatin is a chemo drug, but resistance to it is a big problem. ROS means reactive oxygen species. They can damage cells. And c-Myc is a gene involved in cell growth. So basically, they're trying to figure out how this protein affects how esophageal cancer responds to treatment.
So if they figure that out, could they make cisplatin work better?
That's the hope. If they can understand why some cancers resist treatment, maybe they can find ways to overcome that resistance. Okay, next one. Thioredoxin-related transmembrane protein 1 acts as a prognostic indicator and promotes proliferation and TMZ resistance of lower-grade glioma.
Lots of long words in that one. What's a prognostic indicator?
It basically helps predict what will happen with the disease. Sounds like this protein, if they find a lot of it, could mean the glioma is more aggressive or that it won't respond well to TMZ, which is a chemo drug they use for this type of brain tumor.
So doctors could use this to make better treatment decisions.
Exactly. They'd know which patients are likely to benefit from TMZ and who might need a different approach. Okay, ready for something totally different. Neckband type earphone for continuous monitoring of cardiovascular symptoms via self-powered box knot pulse wave sensor.
Hold on, did you say an earphone that monitors your heart?
That's what it sounds like. And the coolest part, it's self-powered, probably uses your body's movements to generate energy, no batteries needed.
Wow. That would be great for people with heart conditions. They could keep track of their heart rate and rhythm all the time.
Right. Catch any problems early on. So cool.
What's next?
unveiling a novel model of cell senescence-related genes for prognostic assessment and immunotherapeutic insights in gastric cancer.
I think you need a translator for that one.
Okay, so cell senescence, it's like when cells get old and stop dividing. Normal part of aging, but it can also happen when cells are stressed or damaged. And it can play a role in cancer. This research is trying to find genes linked to cell senescence in gastric cancer.
So could that help with immunotherapy? I feel like that's a big area of research these days.
Oh, absolutely. If they understand how cell senescence is involved, they might be able to target those cells specifically with immunotherapy, like get rid of the senescent cells to help the immune system fight the cancer better.
Very cool. Okay, what else?
This one is about implants. Precise, multi-factor, immediate implant placement decision models based on machine learning.
Sounds like machine learning's taking over the world.
Haha, pretty much. So putting in implants, whether it's in dentistry or orthopedics, it has to be super precise. This research is probably using machine learning to figure out the best position, size, all that, for the implant.
Wow, so less guesswork, better outcomes for patients.
That's the idea. Okay, ready for some statistics. Instrumental variable estimation for compositional treatments.
Uh-oh. You know I'm not a stats person?
I'll keep it simple. It's a way to deal with things that can mess up research results. Like, imagine you're studying a treatment, but there are other factors that could also affect the outcome. This statistical technique helps you isolate the true effect of the treatment.
So it's like filtering out the noise to get a clearer signal.
Exactly. Okay, how about a quick trip back to the animal kingdom? Transcriptome analysis to explore the molecular mechanisms involved in the dormancy arousal process in Pomacea canaliculata, Lamarck, 1819.
Pomacea canaliculata, that doesn't ring a bell.
It's a type of snail. And get this, it can go dormant. Kind of like hibernation. This research is looking at how its genes change when it goes into dormancy and when it wakes up.
Snails. Really? What can we possibly learn from that?
You never know. Even though snails and humans are pretty different, we share some basic biology. Maybe this research will help us understand sleep disorders, metabolism, even how to preserve organs for transplants.
Okay, that's actually pretty cool. Nature is full of surprises.
It really is. All right, back to humans. This one is about scoliosis. Genome-wide profiling of circulating microRNAs in adolescent idiopathic scoliosis and the relation to spinal deformity severity and disease pathophysiology.
Scoliosis, that's when the spine curves, right?
Yep. And microRNAs are these tiny molecules that control gene expression. They can fine-tune how much protein is made, which affects all sorts of things in cells. Sounds like this research is looking for microRNAs in the blood that might be linked to how severe the scoliosis is.
So they could be used to diagnose scoliosis or track how it's progressing?
Potentially. Maybe even new treatments down the road. Okay, ready for a slightly creepy one. Pandemic monitoring with global aircraft-based wastewater surveillance networks.
Oh, that does sound a bit creepy, but wastewater surveillance is important, right? It's how we track diseases.
Right, this is taken to a whole new level. They're talking about collecting wastewater samples from planes all over the world, a global network to watch out for new pandemics. Super ambitious, but if it works, we could catch outbreaks early on, maybe even stop them before they become pandemics. Okay, a couple more. How about this one? Predictive analysis of vitiligo treatment drugs using degree and neighborhood degree-based topological descriptors.
Okay, I give up. What is vitiligo, and what are topological descriptors?
Vitiligo is a skin condition where you lose pigment. And those descriptors, basically they're using computers to analyze the structure of different vitiligo drugs, trying to predict which ones will work best.
So more personalized medicine.
Exactly, tailoring treatments to each individual. Okay, last one. Preeclampsia risk prediction from prenatal cell-free DNA screening.
Preeclampsia, that's dangerous in pregnancy, right?
Yeah, it's when pregnant women get high blood pressure. It can be really serious. And cell-free DNA is DNA that's floating around in the mom's blood. Comes from both her and the baby. They're developing a test to see if they can predict preeclampsia risk just by looking at that DNA.
Wow, so they could catch it early and prevent complications.
That's the hope. That's it for Nature.com. I'm telling you, this has been a whirlwind. Plant evolution, the immune system, RNA, machine learning. My brain is full.
Mine too. It's amazing how much research is going on. It gives me hope for the future.
Me too. All these scientists working hard to understand how things work and to find new solutions for all sorts of problems. It's inspiring, really.
I know, right? It really makes you appreciate the sheer volume of research happening around the world.
What strikes me is how everything is so connected now. We saw biology, computer science, engineering, statistics, they're all coming together to move medicine forward.
You need that kind of teamwork to solve really complex problems.
Absolutely. And it's working. We're seeing real progress, better ways to diagnose and treat diseases, a deeper understanding of how things work at a molecular level.
It's exciting to think about the potential impact. All these discoveries could really change people's lives. New cancer treatments, better ways to diagnose diseases early on, personalized medicine, even getting ready for future pandemics.
We're talking about a future where we can detect diseases earlier, treat them more effectively, tailor treatments to each individual. That's the power of all this research.
It feels like we're on the cusp of some major breakthroughs.
I think so too. Science is always moving forward, and who knows what amazing discoveries are just around the corner.
Well, that's all the time we have for today's deep dive. Thanks for joining us on this whirlwind tour, the latest medical research.
It's been a pleasure.
We hope you learned something new and exciting. Stay curious, everyone.